Okay, so good afternoon, everyone, and thank you very much, Tomislav, for inviting me to this kickoff opening and making me place or give me a place to talk about plastic invasion tolling, about some research or data we have gathered studying two lagoon ecosystems in Albania. So I'm trying to share with you today some data on real data on the presence and not only microplastics in the environment, Albanian environments, water mostly, but also about some effects we have seen they have on biota and even on in human organisms. But before I starting to share my presentation, I just a few words about myself. So I, as you can see there, I coming from Department of Biology, Faculty of Nature and Science, University of Tirana. I am a biologist in background and like a special field of research. I'm an ecophysiologist, so trying for almost 25 years to study or to understand the whole mechanisms of how biota or living organisms have or respond to different stressors, including pollution, and in this case, microplastic pollution. So in the meantime, I am also the head of Department of Biology here in University of Tirana, Faculty of Nature Science. And we already just conclude the project on study of microplastics, and these data today are the products of this project. And in the meantime, we have applied for a bigger one, trying to go further into more detailed mechanisms of effect, because nowadays the presence of plastics in Albania, but also in the world, is a fact. So we have it, and we have to understand what are the effects, and in order to find ways maybe to solve this big problem. So yeah, just everyone knows that what microplastics are. So there are small particles, less than 5 millimeters, and the main sources are coming from different sources like textiles, cosmetics, tires, and plastic bags and everything. And every minute it is almost calculated that a size of like a truck, it's already dumped in the waters, including marine waters and also lagoons and wetlands and everything. So as I said at the beginning, it's a very big problem for the moment we are facing at. And here are some, let's say, data where Albania stay on, I mean, among this emergent problem of microplastic pollution. And according to some reports of World Wildlife Fund in 2019, Albania is one of the most problematic countries with the highest percentage of untreated generally waste, but particularly plastic waste. And this ranks it among one of the top 4 countries with the highest norm of untreated plastic waste in the Mediterranean, together with Montenegro, Egypt, and Libya. And also, according to the data of these, let's say, last 5 years, it is calculated that we produce here in our country something like 335 kilograms of waste per citizen, and from these, 9 to 10% are plastics. There have been some efforts to measure, I mean, the plastic, general plastic in Albania. Also, trying to see if there is this plastic is not only in the ecosystems but also in biota. So in 2016, it was a project. It was a Mediterranean project, and it come from the data of the project. It came out that the total marine litter was something about 0.7 kilogram, and 0.2 were found plastics. And if you can see the amount of microplastics present in fish, it was considered to be from 1.65 to 3.67 microplastics per fish. Last 3 years, there are two great projects here running. One is one who tried to see what is the situation of microplastic pollution in Adriatic Sea and fresh waters. And from some preliminary data from them, it is seen that approximately 90% of all marine litter recorded in some north part of wetlands and lakes in Albania were plastic. So it was a big indicator that this pollution is going to be a great and important pollution factor. Also, here are some studies already done, but without going into details with them, mostly they consist of evaluation of marine litter, and very few, for not saying none, until our project came, were focused on the estimation of the presence of microplastics into the biota, and also the potential effects of them into the health of biota and humans as well. So just to go into our study, we selected two big lagoons or lagoon ecosystems in Albania. One is Hunevain, which is an important nature reserve, category 4 from USNA. Then also is a special protected areas according to Barcelona Convent, and also it's an important protected area according to the BirdLife International. So it's a very important ecosystem, and in the meantime, this lagunary system is takes all the runoffs of the two main rivers in Albania. One is in northern part, which is Drini, part of Drini Leja River. So, and he is entering to the adjacent lagoons and communicate with the channels. From channels, sorry, there is a communication between river with the lagoons. And so we were focused in three, let's say, sampling sites, which are mostly indicated for one is the Zaje, which is in the south part, as you see in the map here. And it is because there is a lot of river discharge there. Then we have a Drini Mouth River, which is the main discharges coming from the Drini River, and also the northern part here in Merjan, which is a sampling sites where there is a communication with the sea. So, and the second second sampling site was lagoon of Karavasta, which is also a bigger one. And this is an ecosystem with high ecological value because it's very rich in biodiversity. A lot of wetlands, pines, shallow water lagoons, so it's a complex. And also it's part of it's a Ramsar zone, it's important bird areas and special protected areas as well. So it is a focal point, let's say, for high environmental impact of human or significant anthropogenic introduction. So what we did generally, it was that we looked for presence, and not only, but also we determined the amount of microplastics in water column. Then in two species, the crab species, living there. One is Carcinus estuari, which is a native crab living in the site. And the other one is Calinectes sapidus, which is in, let's say, alien species coming in our waters many years ago, and now it's behaving like, let's say, a common species in the lagoon. But because this one, the second, Calinectes sapidus, is a very big one and very aggressive, and it's very tolerant to the climate changes and everything, high temperatures, and so it's very robust. And so this species is going to it's competitive with the Carcinus estuari, and probably is that they have shifted that from the lagoon. So nowadays usually it's very difficult to find these species in great amounts. Anyway, and then we took also, or did measurements also in the sediments, because we wanted to understand how is the microplastic distributed from water to sediment and to the species. And so the procedure was, I mean, the standard procedure of determining the microplastic in the water, sediments, and organisms. So using peroxide oxidation and then filtrations through very specified nylon mesh. And then we use some incubation with the basic, just to get rid of the all biological elements. And so then we use the stereomicroscopy and polarized optical microscopy at also the FTIR, in order to be able to not only see if there is plastic presence or microplastic presence or not, but also to determine what kind of microplastics it's found there. So I'm showing some data just to have any idea about what we have found. So here are two charts trying to say or to show that what is the abundance of microplastic in water and in the crabs. And what we have seen, okay, there is a variation among the abundance between three sampling sites, and it's understandable if you know the problematic of the place of the sampling sites. But in correlation with the amount of microplastic in water and in crabs, there is a correlation. That means that or it can be understood that the presence of microplastics in water is the same which has been found in the animals, and it is understandable because of the if you know that they are filtratory organisms, as well as they feed on other phytoplankton or zooplankton, so they can biomagnify the concentration of microplastic in their body. So, but when we try to see to fractionate the sizes of microplastic, the most let's say the common size we found is that there was smaller than 1 to 5 millimeters, and the size of microplastics found in the crab and water, it was almost the same. So that means that with a slight difference in the crabs, that we found much more smaller, and maybe it will come from the fact that the crabs took the microplastic not only from water column but also from other organisms. So could be some more degradation of microplastics in even smaller sizes. So or these crabs are all also living in the bottom of the water, so near the sediments. So they took some microplastics also from the sediment, and that's why the concentration, let's say, of the microplastic in the crab's body was greater, and in the continuous Calinectes aphidosauri was bigger because of the size of the animals as well. So there is a correlation between body mass or body weight and the amounts of microplastics found there. Then we try to differentiate between different kinds of microplastics we found. So they almost the same types of microplastics, even in water, sediment, or organisms, going from fragments, microbeads, pellets, and fibers. With, let's say, in water, the mostly dominated type, almost dominated type, were fibers, pellets, and some microbeads. But in organisms, let's say the most bound type found were pellets and fibers. And so there are shown some of the kinds of microplastics, so how they look in microscopy, sterile microscopy, and optical microscopy as well. So they have some microbeads and different sites and different types of microplastic. And if you go for the colors, which are also related with the type of microplastic from the origin. So the most items found were the black ones in water, and the black one in one of the sampling sites, and almost the yellow, etc. So if you're going from the color to the composition of the microplastic, you can understand that mostly of the microplastics come from plastic bottles and packages, because from other studies done on microliter, the mostly plastic waste there were those plastic bottles, bags, and everything we use in everyday activities. So here with TIR, we try to determine which types of microplastics were. So we found there the mostly type found there was this low density and high density, and those polypropylene, and they're the most. So mostly the origin of the plastics found there came from the nets, so nylon from the nets of the fishermen, and the other was coming from the tires of the cars. and the rest was coming from the bags, plastic bags, and bottles and everything throwing by people in the river, or going to the go to the lagoon from the rivers and from other sources. So in one of the sampling sites, which is more near the delta of the river and the point where the lagoon was meet with the sea, there was the highly, let's say, amount of microplastics found, and this is understandable. Then the next step, we wanted, okay, after we found that that microplastic were there and what type of microplastic was, we try to understand what maybe is the effect of microplastics on the other organisms or in the organisms. And because we have a lot of experience in using model organisms like frogs in my lab, we used tadpoles of one common species of frogs here, which is Phelophylax kurtmülleri, because a lot of larvae are found near the wetlands at the small ponds with almost fresh water, where they leave their eggs, and these ponds are transitory, so they use for reproduction. And so we, starting from the amount of microplastics we determined in the water of the lagoon, we exposed the same these tadpole larvae to the same concentrations of microplastics found there. And because we found two concentrations, different concentrations, we used them like 2 doses of microplastics where tadpoles were exposed. And why we use, let's say, amphibian tadpoles like model organism to study the effect of microplastics? For several reasons. Because normally they are sentinel organisms, and they always show are very good showing the problems of the environment when they reproduce. And because they of the style of the life they do, they are exposed to the both ecosystems in land and in water. And because of the fact that they are dual organisms, like have permeable skin, they can be highly exposed to the pollutants. And then they have a high body surface, so they, let's say, this is a parameter that makes or favor the accumulation of microplastics in much more higher rate than in other organisms. And at least because we know very well the physiology and genetic and lifestyle, everything about this species. So you can be able to judge about the effects of a new emergent pollutant can have on this species. And so what we did is that we exposed the tadpoles in the very early stage of their development. So let's say at the Gostner stage, about 25, 27 days, and follow for the whole metamorphosis process. And as well, we keep the control of we have a group like a control in order to judge about the effect of microplastics. So both concentration we use are written there. And what we have seen that the presence of microplastics in the water stressed the tadpoles at that point that their metamorphosis or development either were delayed or even stopped. So at the higher doses, they were never metamorphosed. So they just remains at the larva stage forever. So that means that something happens inside, and when we go further, deeper to understand what happens, we found that there was oxidative stress, higher oxidative stress within the cells. That means that microplastic not even serves like a barrier in the gut of the tadpoles. So by stopping or not leaving the absorbent of the micronutrients in the level of the gut, but even they stressed the cells at a point where the cell goes to apoptosis or let's say programmed cell death. And at the end, so we have some smaller tadpoles or never grow tadpoles. It's very important in terms of surviving on the species. And if we considered another species we have there that is Phelophylax schipericus, which is an endemic one, the consequences are much more harder. I mean, in terms of survival of the species or the status of conservation of the species. Then what happens in the biochemical level? As I told, we found oxidative stress enzymes at very high level. That means that some effects on the endocrine pathways within the organism happens, making the metamorphosis not to go to the end normally. Even there were some damage of the mucus of the or the line endothelium of the gut, and this is also there were all seen by histopathological analysis, and at the same time we have seen also in the level of the blood some damages of the cells. So on the right there it's a normal blood smear from the frog. When you see the typical elliptical erythrocytes with a nucleus in the center, and in the other part you've seen a lot of alterated cells starting from dividing erythrocytes, which is the B in the center of the microphotography, or apoptotic cells, which are C, shown with C in the microphotography, or even immature erythrocytes. All these, let's say, abnormalities, which shows that the effects are much more deeper and harder than we believe. Then we have published these findings of this big project for 2 years project in an article. And now actually we are trying to see what happens with nanoplastic, because okay, it's hard to say that all consequences or all facts I have shown you comes from microplastic itself, because sometimes they are the size of the microplastics is bigger than a cell, for example, but in the environment they can also found in the form of nanoplastic, which is even smaller in size of a virus. So they for sure enter to the cell and cause a lot of damages. And so in cases of persistent microplastic pollution in the water or other aquatic ecosystems, the consequences into the organism could be higher, because for a long-term exposure. And actually, I'm trying to see what happens with exposure of human cells, for example, to the nanoplastic. And for the moment, we have a PhD going on this area, this focus. And we have seen that the nanoparticles size something between 8 to 11 nanometers can enter to erythrocytes and cause a lot of abnormalities, even the apoptosis or the completed cell death. And taking into consideration that in the humans, our cells are not only exposure to microplastic or nanoplastic, but also other pollutants which use microplastic like a support or like a film, like bacteria, for example. So you can imagine that the effect is going higher or more stronger. So we are waiting for other results to say that what other implication could be in the human beings. But yeah, the situation is going to be really alarming. And so there are some conclusions from the project, but as you can see, polypropylene and polyethylene and are the most found polymers, but even nylon as well. So abundance is among, if you compare with other studies in different areas of Mediterranean, are important, not too high, not too low, but still important to get them into consideration. And yeah, microplastic are not longer a future threat. They are already part of our lagoons, but not only, even in Mediterranean and even more wild world. And so our study provides the first evidence of their presence and effects on organisms. And so, and so for the next months and next future months in the future, we will have some data also on effects on humans. So it's important that you started this kind of webinars in order to say or to show with facts that we are not that we are really in danger. I can tell them really it's time to think about to solve this this problem. So this is, I just want to thank my research group and also the partners group from University of Messina and also from University of Pristina and also from Berlin in Germany, when some of our students, when you see here now, are now doing a PhD there. So we try to go further with these studies because we see it as an important threats in not only in Albania, but as I said, in the whole Mediterranean and even wider. So yeah, thank you.